S, and together it gives 70S. S over here is the sedimentation coefficient, and it's also termed as Swedberg's unit. It is indirect way to assess density and size.
Yes, right.
Yeah. And while in eukaryotics, the cell has 60S and 40S, which together gives 80S. And
Yeah, but this does not adds up, right?
Yeah, this does not add up.
Why? Remember we discussed?
Yeah.
Yeah. Because Aisha can ask that 60 and 40 does not become 80. Yeah.
This is
So
because
for the next 10 minutes, Iram is the teacher for Aisha. Okay. So Aisha, you can ask questions if you want. Yes, Iram.
the 50S and
Pressure.
30S gives
Iram feels the pressure the moment I say that she's a teacher. No pressures, okay? Relax. You're doing good.
Okay. So 50S and 30S does not give 80S because here the units are not being added up. It's the sedimentation rates, right? So
No, it's not also the sedimentation coefficient. We are not talking about the rate here. We are talking about the coefficient, okay? It's just a number, just like any other coefficient. You have been using many coefficients in physics, right? Just to make an equation work or make an equation balance an equation, we add coefficients, right? This actually tells us, remember I used two things in the last class. Let me use some other things again, if I may. I don't keep much things on my study table, I realized. Okay, but anyways, can you see these two things? Right? People? Yes. So one is bigger. This is the bigger subunit of the ribosome. This is the smaller subunit of the ribosome. So let's say for a eukaryote to make 80S, this is 60S and this is 40S. Okay. But as the diagram says here, look at this diagram here. The bigger unit and the smaller unit are attached together. So the bigger basically sits on the top and the smaller is at the bottom, right? This figure is also in your NCERT. So when the bigger subunit sits on the top of the smaller subunit like this, some surface area, some portion of both the subunits gets hidden. You understand? And now together this becomes one molecule, or sorry, this becomes one bigger entity. Now the coefficient rate, sorry, sedimentation rate of this bigger unit will not be the exact sum of the sedimentation coefficients of these two smaller units, because here the surface area for each was different. This was 60, this was 40, but when they merge together, the total instead of becoming 100 is 80, because 20 got hidden. You understood? So this is not exposed and not playing a part in the sedimentation now. Do you understand, Iram and Aisha?
Yes.
Yep. Okay, very well. Continue, Iram.
yes, so yeah, ribosomes are done. The next part is gram staining. We discussed about gram staining. So on the basis of cell wall, bacteria can be classified into two: gram positive and gram negative. Okay, so gram is basically named after the scientist Christian Gram, and it's gram staining is basically coloring or staining of the cell wall. Okay. So first I'll talk about gram negative. Gram negative, gram negative cells, they are surrounded by a thin layer of peptidoglycan, and over it is also surrounded by a layer of lipopolysaccharide. Okay, and gram negative bacteria, it's surrounded by a thick layer. Okay, a thick layer of peptidoglycan. They do not consist of lipopolysaccharide. So when you add the gram stain, which is of iodine, crystal violet, ethanol, the stain is of. When you add the stain to the cell, if the cell turns blue or violet, it is called as gram positive bacteria. And if the cell wall does not retain the color, means it does not turn blue or violet in color, it is called as gram negative bacteria.
Yep.
Correct?
Yes, right. And also, I forgot to tell you something in the last class. There was one question in the exam which of the two, gram positive or gram negative, is more harmful and is difficult to kill using antibiotics? You know, so antibiotics basically affect this peptidoglycan layer of the cell wall. Okay, so if something has a thicker, if the cell wall is only made up of peptidoglycan and no lipopolysaccharide is there, it is easier to kill this bacteria through antibiotics, right? Because you degrade this and there is no layer of protection now. But in case of gram negative, you have peptidoglycan, which is very thin. So even if you degrade or you damage some bit of peptidoglycan, there is a lipopolysaccharide that you cannot damage. So because of the presence of this lipopolysaccharide, gram negative bacteria become more harmful and are difficult to kill. Okay. So just write down, gram negative bacteria are more harmful and difficult to kill due to the presence of lipopolysaccharide layer.
Gram negative or gram positive?
Gram negative. Okay. Gram negative is difficult to kill. Gram negative bacteria are mostly present in, like, there are some non-lethal ones as well, but again, they are difficult to kill. Even if they are not causing a disease, they are difficult to kill because the lipopolysaccharide is like, it's like a, as the word says, lipo, it has lipid in it. It's like mucus layer on the top. In our body, we have, wherever we have lipids and we have mucus, what is the purpose of that? Where do we have mucus in our body? Mucus, people. Everywhere, the whole internal lining of our body is covered with mucus. The nasal cavity, the mouth, the food pipe, the windpipe, the lungs, even the stomach. Stomach has a very thick layer of mucus, which prevents the stomach from HCl, right? You know that? You must have studied this.
Yes.
Yeah. Then in the intestines, large intestine, we have mucus all around. It serves two purposes. It lubricates the internal lining. It also protects the internal lining. So similarly, these kind of structures present in the cell wall of a bacteria protects the bacteria, give additional advantage to the bacteria over the gram positive ones, which only have peptidoglycan. Okay. Iram and Asha?
Okay.
Okay. Okay, let's continue. Iram.
Yeah.
Then we started with eukaryotic cell.
Yeah, in eukaryotic cells, we discussed about endomembrane system. Endo over here means inside, and membrane system means membrane system. So, endomembrane system, it comprises of 4 cell organelles, which are ER, Golgi complex, lysosomes, and vacuoles. So these cell organelles inside the cell functions in a coordinated manner, and this brings about the endomembrane system. So first we'll start with the ER, endoplasmic reticulum. Endoplasmic reticulum, it is an extensive network of tubular structures. At one end, it touches the nuclear membrane, and at the another, it touches the cell membrane, or the plasma membrane. And there are two types of ER. One is the rough endoplasmic reticulum, and another is the smooth endoplasmic reticulum. So rough endoplasmic reticulum, ribosomes are attached to the outer surface, due to which it is called as rough endoplasmic reticulum, and their function is protein synthesis. Whereas smooth endoplasmic reticulum, they do not consist of ribosomes, due to which they appear smooth, and their function is lipid synthesis.
Yes.
Okay. Now, the second organelle is Golgi apparatus. Golgi apparatus was discovered by Camillo Golgi in the year 1898. It is a reticular structure near the nucleus, means it's near around the nucleus. And Golgi, it's like a disc-like structure connected together, and each disc-like structure are called as cisternae. There are two faces. You can see one face is the concave face, one face is the convex face, which is called as the cis face. It is the forming phase, and another phase is called as the concave phase, trans phase, and it is known as the maturing phase. Now, functions of Golgi bodies, it packs things and materials that are to be delivered intracellularly and intercellularly. So the word intra means within the cell, and the word inter means between the cells. Yeah. So, for example, if materials produced from ER
Intra
are to be
means
packed, the word
Yes.
intra means within the cell.
Yes. So, Aisha, you must have heard intra-school competitions and inter-school competitions, right? There is some
yes.
inter-school debate competition. So the inter word means between two schools, school A and school B. It's inter state, international. So the word international means between many countries, right? Intra is within the same thing. So if it is intracellular, it's within the cell. It's not, it will not go out the boundary of the cell. Okay, inter and intra. So
Yes.
both intracellular delivery and intercellular delivery is packed by Golgi. And I think Iram just missed one point here, that anything that enters the Golgi for packaging enters from the cis phase. That's why it's called the forming phase. And as it moves from the cis towards the trans phase, it becomes mature. Whatever the substance is, it becomes packed, labeled, and mature. And then the mature thing, which is packed, is released from the trans phase. Okay, that's why it's called the maturing phase. Yes, continue.
Yeah, so anything, so the material produced from ER, which are to be packed and transferred to other cell organelles. So first it goes through the Golgi bodies. It enters the cis phase, which is the forming phase, and move towards the trans phase. Okay, and
Yes.
Golgi also remains in close association with ER. It is besides that, it is also very important for it is an important site for the formation of glycoproteins and glycolipids. It also helps in the formation of lysosomes.
Yes. Right.
Now the third cell organelle is lysosome. Okay, so they are membranous structures that are formed and packed by the Golgi apparatus. Lysosomes are also called as the suicidal bags. They These contain strong hydrolytic enzymes of all the types. Example, it has lipases for the breakdown of lipids, proteases for the breakdown of protein, amylases for the breakdown of carbohydrates, and nucleases for the breakdown of DNA or RNA. Yeah. Now we have the last, vacuoles. These are membrane-bound spaces present in the cytoplasm. They are just like the storerooms. Basically, anything, any waste material or anything which has to be stored just goes inside it. And yeah, yes,
No.
yeah.
And what's the special thing about vacuole membrane? Remember tonoplast?
Yeah. See. So it has, you can see it has this membrane. It's a single layer of lipid membrane. It's called as tonoplast. So vacuoles mostly store water, sap, waste, waste. And in animal cell, they are multiple tiny vacuoles, whereas in plant cell there is a single large central vacuole that occupies up to 90% of the cell space. So the function of the tonoplast is that once a material is put inside a vacuole, even though the concentration gradient of that substance is more than that of the cytoplasm in the vacuole, so it would prevent it from the backflow, right? It won't
Yes,
allow the
right.
movement
That's
of the
that's the
material out of
speciality of a, yes, that's the speciality of a lipid monolayer, and that's the intelligence of the system, that we have lipid layer at some other place as well. Remember cell membrane? Cell membrane is a lipid bilayer. It has two layers. That's why through protein channels or just through diffusion against the concentration gradient, you can make the things move in and out from a cell membrane. But in vacuoles, whenever plant or animal puts something in the vacuole, most of the time it is useless substance. Okay, very few examples are there where we use vacuoles for useful things. And in that case also, where we use it for useful things, we do not want the movement to and fro, inside out. We just put things in the vacuole and never take it out. Either we just remove the whole vacuole, we do not need it anymore, or we digest the vacuole. But it very rarely it happens that what goes inside the vacuole is required again in the cytoplasm, and it has to come back in the vacuole. So this tonoplast, which is a lipid monolayer, helps in the transport of substance against their concentration gradient. So you can keep pushing as much as you want. You just need to make bigger vacuole. And suppose you want to put all the sap in the, or all the waste material in the vacuole. So the concentration of that waste material will be very high inside the vacuole and will be very low in the cytoplasm. But again, the next time when there is a waste produced in the cytoplasm, you can still put it in the vacuole. Okay, that movement is still possible against the concentration gradient. Otherwise, normally it should have been coming out of the vacuole now, right? Once there is a balance of the concentration inside and outside, then the motion stops. Then wherever the concentration rises, the motion happens in that direction, right? Normally through diffusion.
Yes.
But here it's not happening because vacuoles are special. Yes, and there were some useful as well. Yes, let's end.
And there's also a surprising fact that gold and silver nanoparticles are also stored
yes.
in the vacuoles.
Yes, we were discussing this last time, though it's not in your syllabus, but I was telling Iram that there are certain plants which do not know what, so they can imbibe, or they can actually absorb certain gold and nanoparticles and other metal particles as well. So why I was just telling about gold and nanoparticles, because normally plants keep taking other like copper, iron particles, but copper and iron is not something which is interesting to talk about. So they can also take up gold and silver, and they do not know what to do with it. So they will put it in the vacuole, the large central vacuole. And if a plant lives for 100 years and it is like hundreds of feet long, then in every cell there is at least some molecules, some nanoparticles of gold. And if you just take that total weight, one plant can hold up to half kg or 1 kg of gold in itself. But the point is we cannot extract it. And I think it's a good thing that we cannot extract it. Otherwise, humans would have brought down all the plants to extract gold out of it. Yes, that's interesting. So plants can technically put anything in the vacuole, anything which they can put there. Yes.
And they're
I also told you, Iram, we were discussing that gold and nanoparticles are used as immunity boosters. Remember?
Yeah.
We eat gold and silver coated on the mithai or the sweets, right? On kaju katli, something like that. Yeah. Did you go back and figure out how does it help us with the immunity? Even Chamanprash has gold, and all these commercials say that it has gold and silver active nanoparticles. I'm not sure if they do put it or not, but they say it because they know that it is an immunity booster. Anyways, we'll talk about it. Let's finish with useful vacuoles first.
Yeah, they are also useful vacuoles. Food vacuole and contractile vacuole. Food vacuole, it is formed by engulfing a food particle. Example, amoeba, it forms a vacuole while engulfing food. Second is the contractile vacuole, which helps in osmoregulation. It is regulation of osmosis. You can say diffusion of water inside of the cell, and it is also present in amoeba.
Yes. So is it clear, Aisha? Everything is clear to you?
Yes, sir.
Yes. Okay. So with this, we finish the endomembrane system. There are 4 components of the endomembrane system. The ER, endoplasmic reticulum, both rough and smooth, the Golgi, the lysosomes, and the vacuoles. So these 4 make the endomembrane system. Now let's come to those organelles which are not a part of endomembrane system, but are very, very important. Non-endomembranous organelles. Okay. Now in that, I would like to start first with the after nucleus, the most important organelle called mitochondria. You know what you know about mitochondria from junior classes?
So,
Yes,
Yes, there was a question that you gave in the previous, not in the previous, I think 2 classes back. It was
Mhm.
that which organism has calcium carbonate in the cell wall?
Yes, which one?
It is
Could you
algae.
Algae.
some, but have you heard of diatoms and desmids? Diatoms have silica also in their cell wall. So silica, and have you heard of corals, coral reef? Coral reef, they are very beautiful looking designs. They look like plants actually. Have you seen any underwater movies or any movie like Finding Nemo? So these fish, when they live around some beautiful colorful things on the rock, they are called corals, and the word is called coral reef. Do you know about this? You're right, some algae also have it, but corals, desmids, they have calcium carbonate in their cell wall, and that is what make them so resistant and strong. So you cannot, it's not very easy to kill these organisms. Okay, all algae do not have calcium carbonate. That's the point. Hiram, is it clear?
Yes, clear.
Yeah. Okay. Now, tell me, what do you know about mitochondria from the junior classes? At least the famous name that we give to mitochondria, it is called the
Powerhouse of the cell.
powerhouse. Why do we call it the powerhouse? Why do we call it the powerhouse?
It has its own DNA.
That's, it has its own DNA, but
It
that's not why we
produces
call it.
ATP.
Yes, it has the ATP synthesis mechanism, or the process of ATP synthesis happen here, or we call it aerobic respiration. Without mitochondria, a cell cannot do aerobic respiration. That's why mitochondria is not present in bacteria, right? But bacteria also do respirations, right? What is
Yes.
respiration? What is respiration? Respiration is a cellular process, okay, where we break down glucose in the presence of oxygen, right, to produce energy. Now that process is called glycolysis. You know about that? It happens in cytoplasm. It does not require mitochondria. So glycolysis happens in all organisms, from bacteria to human. But why are we better than bacteria? Because from the same amount of food that we eat, we can make more energy, because we have something called mitochondria, where we can send the end product of glycolysis, and we can produce even more energy from it. Okay, multiple times more energy than a bacteria. That's why it's called the powerhouse of the cell. So first thing, write down, mitochondria is a double membrane bound structure. So some characteristic features, it is double membranous. What other thing is double membranous in the cell? Can you think of anything else? Any other organelle?
Plastids.
Yes, and you forgot the king of the cell, the brain, nucleus. Nuclear membrane is also double membrane. Okay. So mitochondria, the similarity between mitochondria and nucleus is both are double membranous, and what Iram said before, both have their own DNA. Nucleus has its own DNA, and mitochondria also has its own DNA. So write down, it's a double membranous structures with the outer membrane. Now normally the difference is, in the nucleus, the inner membrane is smaller than the outer membrane. Makes sense, right? But in the mitochondria, the inner membrane is bigger than the outer membrane. Now, how can a bigger membrane fit in a smaller membrane? What is the only way?
By folding it.
Exactly.
foldings.
Yes. So you have to induce foldings if you want to accommodate a bigger membrane inside a smaller membrane. And this is exactly what mitochondria do. But why is mitochondria doing this? Is the question. Why not like nucleus have a smaller membrane inside and a bigger membrane outside and live peacefully? Why you need to do all this hustle of maintaining a bigger membrane inside and a smaller membrane outside? Any idea? Because if you understand this, it becomes easier for you to understand the purpose of mitochondria. Because let me first tell you everything about mitochondria, then I'll dictate. Because the production of ATP, the ATP synthesis, the energy producing mechanism happens in the inner membrane. So all these proteins that do this business are embedded in the inner membrane. They are not on the outer membrane. So that's why to increase the surface area of the inner membrane, so each mitochondria being smaller can work more than its own size. Mitochondria maintain an inner membrane which is bigger in a folded state, okay, to increase the surface area. And the inner membrane, between the outer membrane and the inner membrane, there is space, okay, and that space is very important for the function that I'm going to tell you now. Okay, so write down first. Mitochondria has an outer membrane which has lesser surface area and an inner membrane which is folded and has greater surface area. And if I draw it as a structure, let's say if this is the mitochondria and this is the bigger inner membrane, so it will be like this, right?
Yes.
You can see that it's very, very long as compared to the outer membrane. If you just stretch it out, it will be bigger. But because of this, you now see that there are two spaces. One space is this one between the outer and the inner membrane, right? And one space is inside the inner membrane, right? So the lumen is internal space of the mitochondria. So the lumen is divided into two spaces distinctly. Now write down the inner compartment. So this is called the inner compartment. Inner compartment. The inner compartment or the matrix. It has a dense inner compartment, has a dense substance called mitochondrial matrix. Is it clear? And these foldings which you see here in this inner membrane, these foldings have a name. They are called cristae. Do not get confused between cisternae, which were the folds of the Golgi, and cristae. Cristae are the folds of inner membrane of the mitochondria. Okay. In foldings of inner membrane. Cristae, inner compartment. This is the outer compartment. Next point. Both these compartments, inner and the outer, write down, both the inner and outer compartments have their distinct or their different enzymes. That's one thing. And mitochondria, in biology, there are many questions that come which are site based. So what is the site of this? What is the site of this? So if they ask you what is the site of glycolysis, it is cytoplasm. What is the site of lipid synthesis? It is quick, Iram. What is the site of lipid synthesis in a cell? Smooth endoplasmic reticulum. Remember? Am I audible, Iram? Yeah. What is the site of aerobic respiration? Then it is mitochondria. Write down. Mitochondria is the site of aerobic respiration. Aerobic respiration is which requires oxygen. You must have studied aerobic and anaerobic, right? I do not have to explain that. Both are clear, Iram and Aisha, with aerobic and anaerobic respiration? Okay, very good. Now what happens, write down, site of aerobic respiration is the inner membrane. What happens is they produce cellular energy. They produce cellular energy, which is also called as chemical energy, known as ATP. Now, do you know what is a ATP? What does ATP stands for? It is not in NCERT, but still it will be. It can be asked.
Adenosine triphosphate.
Yes, it is called adenosine triphosphate. Now, this triphosphate word has all the energy. Now it looks like this. So there is a big molecule called adenosine. I'm not going to make the structure of adenosine. So just know that there is a big molecule adenosine carrying 3 phosphates. 1, 2, and 3. Okay. The second and the third phosphate, first phosphate, second, and the third. The bond between the second and the third is called a pyrophosphate bond. Why it is called the pyrophosphate? What does the word pyro means? How many of you know what do we take when we have fever? You both must have had fevers some points at some time of your life, right? Fever is a just defense mechanism of the body. If you have any inflammation in the body will induce fever, right? What do we take to control fever?
Panadol.
Yes, there are many names for the local names for the medicine, but you know chemically what that medicine is called? What do you take for pain? No, it is antibiotic is which kills bacteria or any other life form. Anti is against, biotic. Okay, let's come here. Antibiotic. The word bio means life. Antibiotics means against life, which kills any life form. Now this life form, antibiotics are of various kinds. If we are killing bacteria, it is called antibacterial, antifungal, antiviral. Okay, all are antibiotics. So they are mostly we use it against bacteria. What do we use for pain? Forget pain. What do we use for fever? Those are called antipyretics. Paracetamol, Dolo, or whatever you call, they are called antipyretics. They are against, the word anti means against. Pyretics is against the high temperature or fever. You understand, everyone? Iram, Aisha?
Yes.
Can you see the word pyro or pyre here? Same word is here. Pyrophosphate bond. Now, what can you understand from this bond? This is the bond which contains the energy. So if you have ATP in the cell, you have energy stored in the pyrophosphate bond. Whenever you need the energy, you just break this bond, convert this ATP into ADP, adenosine diphosphate, plus one phosphate, inorganic phosphate will be released. So ADP plus phosphate, because this bond got broken and energy got released. So whenever we need energy for doing anything in the body, even to smile or to frown or to walk or to do anything, we break ATP into ADP and phosphate and release that energy, which we can use. Now this energy is released in the form of heat. Okay, that's why the word pyro comes. Heat energy is released. So when you break this pyrophosphate bond, heat energy is released. Do you understand? And that's why when we have fever,
Yes.
to bring down the temperature, we use antipyretics that can bring down the temperature. Right. So this ATP is synthesized in mitochondria. ATP synthesis happens in mitochondria, and ATP is also known as energy currency of the cell. This is the currency that is valid in the cell for all transactions. Okay, energy currency of the cell. Now, coming back to mitochondria, write down. Mitochondria contains its own genetic material, its own DNA, and also codes for contains its own DNA also codes for some mitochondrial proteins. Okay. Now this DNA is a single circular DNA. It is not like a thread in our nucleus. It is single and it is circular in a loop. And if it has its own DNA and if it codes for its own proteins, do you think that mitochondria should also have its own ribosomes? Yes or no?
Yes.
Yes. So
Yes.
do you mean to say that inside an organelle there is another organelle? Because ribosome is a legit organelle in our cells. So we have ribosomes attached to the ER and also freely present in the cytoplasm. Bacteria also has ribosomes in its cytoplasm. Now you mean to say in a eukaryotic cell, there is ribosomes of the cell, there is mitochondria, and there is ribosomes inside the mitochondria, which is mitochondrial ribosome.
Yes.
Yes. So let me ask you, what will be that ribosome's S unit? Will it be a 80S ribosome or a 70S ribosome? Because the cell is eukaryotic cell.
ETS.
Mitochondria is only present in eukaryotic cell, right? So technically, we will think that any ribosome in the eukaryotic cell must be a ATS ribosome, correct?
Yes.
Mitochondria contain their own 70S ribosomes. Write down. Its own ribosomes. These are the ribosomes which mitochondria uses to synthesize protein from its own DNA. 70S. Normally, 70S ribosome is present in which organism?
Bacteria.
Exactly. Right, correct. So mitochondria has 70S ribosome. Mitochondria has a single circular DNA. Mitochondria also has, write down, contains some RNAs, its own RNAs. Okay. Now that's not all. The interesting part is yet to come. Mitochondria is self-replicative. It can divide, one mitochondria can just divide and form two mitochondria through binary fission. Now read all these 4 points again and tell me, do you suspect mitochondria is not behaving like an organelle and it actually is behaving like a bacteria? Yes? Yes, Aisha is convinced, but I think Aisha is
Yes,
thinking,
yes.
what is happening here? What is this mitochondria up to? So mitochondria is supposed to be an organelle in our cell, right? In every eukaryotic cell. But it is, the characteristics tell that mitochondria is actually behaving like a prokaryotic organism inside a eukaryotic cell. Go back to prokaryotic versus eukaryotic diagram. Let me go back there. What do you see here? Inside a prokaryotic cell, there are ribosomes. Yes?
Yes.
Inside a prokaryotic cell, there is a single circular DNA.
Yes.
Yes.
A prokaryotic cell like bacteria divides by binary fission.
Yes.
Yes.
Yes. And inside a prokaryotic cell, there is also the mRNA which is produced from this DNA. And then those mRNA are read by the ribosomes and proteins are produced, right?
Yes.
Yes.
So mitochondria, being an organelle, actually resembles like a prokaryotic organism. Yes or no? It has its own genetic material, own ribosomes, own RNAs, its own way to divide and form more copies, and its own circular DNA. Yes?
Yes.
So this is where it is proofs that mitochondria at one point of time, when cells were evolving, when the first eukaryotic cell evolved, the eukaryotic cell, actually one bigger cell, which became eukaryotic, engulfed another smaller prokaryotic cell, which were the precursors of mitochondria. So mitochondria are technically, biologically, they are aliens. They are other organisms living inside our own cells, but they have accepted our cells as their home, and they started helping us to produce more energy, and we started helping them for shelter, nutrition, and a safe environment to multiply. Amazing, right, Iram? Yes. I was just waiting
Yes.
for that emoji. I was just waiting for that emoji to come. Yes, it's from both now. And I was waiting for this one also. So this actually, this whole theory, where did mitochondria? Because when scientists started studying mitochondria, they were amazed. They thought, how can an organelle which is developed inside the cell, if something evolved inside the cell, like endoplasmic reticulum, it evolved inside the eukaryotic cell, like Golgi body, it evolved inside the eukaryotic cell. So when it evolved inside the cell, it cannot have its own genetic material. It will rely on the cell's genetic material, the nucleus, right? All the commands will come from the nucleus. It will only have the structure which is important for the function for which it is being evolved. But if an organelle has everything of itself and still is inside our cell, probably it did not evolve in our cell. It came from outside in eukaryotes and then started living with a eukaryotic cell. And this is actually theory which is called endosymbiotic theory. Endosymbiotic theory. Write down, you have to go back and let me just, yes, write down endosymbiotic theory for mitochondria. Just like mitochondria, there is one more organelle which has its own DNA, its own ribosomes, its own RNAs, and a double membrane. Iram just took the name few minutes ago.
Plastids.
Chloroplasts, right? You call it chloroplast. Plastids are present. So chloroplasts are also like mitochondria. They are only present in plant cell. Mitochondria is present both in plant and animal cell. But endosymbiotic theory for mitochondria and also for chloroplast is there, and which has some proofs to believe in. Okay. Otherwise, it does not make sense for an organelle to have all of these characteristics, right? Why do you have your own ribosomes? And that also 70S ribosome. 70S ribosome is a characteristic property of
Prokaryotic cells.
prokaryotes. So why do you have a 70S ribosome when you are living inside a eukaryotic ribosome? Living inside a eukaryotic cell. So if you were evolved, if you evolved in a eukaryotic cell from the beginning, this 70S ribosome could not have been a part of yours system, right? Even if I could still believe that mitochondria did not came from outside, it evolved in the cell only, and all these things it took from the cell only. Then 70S ribosomes cannot be present, right? Because eukaryotes do not make 70S ribosomes. Yes or no?
Yes.
Even the subunits are different. 60-40 makes up 80S, and 50 and 30 makes up 70S. So there is no way to make a 70S ribosome in a eukaryotic cell, but mitochondria does that because it has its own ribosomes from the beginning, from where it came into the cell. So this is the theory endosymbiotic. The word endo means inside, symbiosis means living together. So two cells, one prokaryote, one eukaryote, decided to live together in a mutual relationship where the smaller one will get shelter, protection, and nutrition, and the bigger one will get energy from that smaller one. So the smaller one, which is mitochondria, said that let's sign a deal. I have a very big membrane. I'm double membranous. I have a bigger, smaller membrane, and my specialty is that I can do aerobic respiration. So you give me the raw materials. I will do the aerobic respiration. I will give you lots of energy that you can use to maintain yourself. Then you provide me with whatever I need: shelter, protection, and nutrition. Done deal. Good. Both are happy. And from that time, millions of years ago, this happened. And today, it's known as mitochondria, our powerhouse. How do you feel about it? Good, bad, happy, glad? Iram feels good. Aisha is still surprised. Okay, cool. Now let's come to the closer counterparts, which are plastids. Tell me, what do you already know about plastids? Now you will tell me the same story again. You're right. What are basically plastids? What is the difference between a plastid and a chloroplast? This is my question. Are both same? Just give me 1 second, okay? I'll be back. Think about this question. What is the difference between plastids and chloroplast? I'll be back in 30 seconds. Earlier than I promised. Okay. Yes, Iram. What are chloroplast? What are plastids? Are they both same?
No,
Yes,
they
no?
are.
They are different? What is the difference, Aisha or Iram? What is the difference?
Chloroplast can be green in color,
Not can be. Chloroplasts are green in color.
are green in color.
If they are of some other color, they are not chloroplast. Okay, that's the
Yes.
difference. All chloroplasts are plastids, but all plastids are not chloroplasts. Okay, first rule. It's like everything that glitters, like gold always glitters, but everything that glitters is not gold. Okay, so plastids can be of multiple types depending on the pigments that they carry. If a plastid is carrying green pigment, which is chlorophyll, A, B, or C. So chlorophyll containing plastids are called chloroplast. Chloroplastids. The word is, a kind of fusion. Chlorophyll containing plastids is a long term to say, so they said chloroplast. Make sense, Iram and Aisha?
Yes.
The ones that contain other pigments are called chromoplasts. So plastids, the plast word comes from plastid. They can either be, the most common are not chlorophyll containing plastids, chloroplasts. Second one, they do not have green, they do not have chlorophyll, but other pigments like yellow, brown, xanthophyll, etc. They are called chromo. The word chrome means chrome, or chromo means color. Okay, the word chrome, monochrome. You all use filters, right? Monochrome filter, Snapchat, Instagram, come on. What does the word mono means? Everything is science. Yes, Aisha. Aisha knows. Oh, yes, I use monochrome filters. What is a monochrome? What does the word mono means? Single, one, right? Chrome
Yes.
means color. So any filter that only enhances or is based on one color is called a monochrome filter. Okay. So chromoplast is other colors. Plastids containing other colored pigment apart from the green pigment, which is chloroplast. The third one is called leukoplast. What does the word leuko means? Leuko.
Storage.
No. What is the scientific name of your white blood cells? Leukocytes. White blood cells are not white actually, it's a misnomer. But yes, the word there stands true. The word cyte is for cell. The word leuko is for colorless. The word is colorless. Have you heard of this one disease called leukemia? Say yes or no.
No.
Yes, you have. Iram, you say yes or no. First say yes or no, then I'll tell you the other name for this disease, and again Aisha will give a laughing emoji. Leukemia is commonly known as blood cancer. Have you heard of this disease, Aisha?
Yes.
See, I bet that you know. So in leukemia, the leukocytes grow in number because blood is a tissue. It's the only liquid tissue in the blood, in the body. So when the number of blood cells, the leukocytes, keeps on increasing to a level where they reach saturation, that's called leukemia or blood cancer. Okay. So leukoplast, this was all to tell you that leuko means colorless. Leukoplast means those plastids which do not contain colored pigment, they are colorless. Okay. So if they are not containing any pigment, what are they containing? Something else, right? So let's go to all of them one by one. Chloroplast, write down. Oh, sorry. Mistake. Chloroplast contains chlorophyll, which is green colored pigment, which helps in which process does it helps in?
Photosynthesis.
Yes. You know what does the word photo means?
Light.
Everything is science. Great. And what does the word synthesis means?
Producing.
Yes. So you are producing something in the presence of light. What are you producing? You're producing food, nutrition. So that's called photosynthesis. So with the help of chloroplasts, plants and algae can harness the energy of sun, energy of light, from physical to chemical energy, and then use that chemical energy to produce nutrition. Okay, that's called photosynthesis. Cool, everyone? Coming to chromoplast, write down. Chromoplasts, or let me first give you a brief, then I'll dictate. Chromoplasts are like, they contain pigments like carotenoid. Carotenoid. You know where is this present? Carotenoids? In carrots. That's why they are called carotenoids. Okay. They are non-green pigments, orange, brown, blue, all these sorts. Leukoplasts are colorless. Okay, they do not contain pigment. So what they contain? They contain either carbohydrates or they contain fats or lipids. Okay, fats, oils, or lipids. So if they contain carbohydrates, they are called, what is the another word for carbohydrates? You know, do you know what is present in our mouth? An enzyme that dissolves or
Amylase.
digests.
Saliva.
Yes. What is present in saliva?
Amylases.
Amylases. So amyl, the word amyl is used for carbohydrates. So if it contains carbohydrate, what will be the name of this plast? Sorry.
Amyloplast.
Perfect. Eram. Was it Eram, right?
Yes.
You're already good at it. Amyloplast contains amyl carbohydrates. And if they contains lipids and fats, what they will be called? Elaioplasts. Okay. So chloroplast, chromoplast, leukoplast, amyloplast, elaioplasts, all are clear?
Yes.
Okay. Now let's come to the plastids, the basic things about it. So start writing.
So
Yes.
these leukoplast also consist of. No, these also stores protein, right?
Yes, right. Forgot that. They are called
Aeroplast.
aleuroplasts. They are also protein storing leukoplasts. They're called Aleuroplasts. Yes, thank you, Ira. Now let's come to plastids. Write down. Plastids are present in all plants, all plant cells, and also they are present in euglenoids, euglena. You know euglenoids? You know euglena, an organism, euglena? No?
Yes.
Yes, okay, then good. Otherwise, go and figure out euglena. It's like,
Type of algae, right?
sorry?
It's a type of algae.
Euglena is actually at the borderline of being a plant and an animal. It contains chloroplast, so it can behave like a plant when there is sunlight available. Okay. Algae are proper plants. We put euglena in algae, yes, like for study they are in algae because they are eukaryotes and they have a flagella. All the algae do not have flagella, but euglena have a flagella. You know what is a flagella? A long, in bacterial cell there were flagellas, right, which help in motion. Normally all algae do not have flagella, but euglena has chloroplast, chlorophyll and chloroplast. That's why it's put in algae. You study it as an algae, but it can behave like animal also. If it is dark and you cannot use chloroplasts to produce energy, then euglena can use the flagella, move around, find smaller organisms, eat it. Okay. Is it clear, everyone? Yes, no?
Yes.
Iram, Aisha? Yes. So it's present in plant cells and euglenoids and plastids, and they can be of three types. Then just write this down. Just make this chart, this flow chart. Let me know when you are done making this flow chart.
Done.
Done. So they can be of three types. The chart is done. Now write down.
No, sir, I'm doing.
Okay, do it. Let me know when you're when you're done, Aisha.
Sir, done.
Done? Okay. Now coming to structure of plastids. This is where many questions come from. Now, just like mitochondria, plastids, chloroplasts, chromoplasts, all plastids also are double membranous. Okay, so write down. First thing, they are double membranous. Now here the arrangement is slightly different. Here the inner membrane is not bigger than the outer membrane. Okay, because here whatever is happening, the activity, the chemical activity is not happening in the membrane. They have another structures for it. So if am I audible? Okay. What happened? Am I audible, Iram, Aisha?
Yes, yes, it's audible.
Okay. So let's say this is the outer membrane for plastid. And then there is a inner membrane. Inner membrane is just like the nuclear membrane, smaller than the outer membrane. This is the difference between plastids and mitochondria. Okay. Between the outer membrane and the inner membrane, there is a very less space. The outer membrane is permeable, actually semi-permeable, but it is permeable to many things. It can allow many things to exchange, but the inner membrane is less permeable. It does not allow things to go inside or come out very easily, because of which the internal region, this part, the space within the, write down, the space enclosed by the inner membrane is called stroma. Let me first draw it. You just listen and understand, then I'll give you time to draw. Space enclosed by inner membrane. Okay. Now in this stroma, there are enzymes present. And these enzymes are actually present, which they synthesize carbohydrates. Carbohydrates and proteins. Now, do you understand where does that carbohydrates in amyloplasts and protein in Aleuroplasts come from? They synthesize it inside the stroma. Is it clear, Iram and Aisha? This part is clear?
Yes.
Yes.
Yes. Yeah. Now in this stroma, I'll use another different color now. Let me use this one. In this stroma, there are structures which are arranged, like they are disc-like structures which are arranged like coins, one on the top of other. Okay. So just like you stack coins one on the top of other, they are disc-like, round. There are many. I will make just 3 stacks to explain. These structures are called grana. A single one is called granum, and the complete stack is called grana. Multiple, they're all grana. One stack is called granum. If you take both 2, 3 stacks, it will be called grana. Is it clear? So you understand what is grana? Second thing which is confusing is the word thylakoid. So you know this grana is the pile. Many coins, one on the top is together called, that pile is called grana. Multiple pile is called granum. But this one disc which is there, the membrane of this one disc is called thylakoid membrane. Do you understand? So multiple thylakoid membranes stack together to form a granum. Multiple granum stacked together called grana. Is it clear? Yes, no?
Yes.
These individual granum are connected to each other. Okay. Now this connection with which they are being connected here, this is called let me not put this here. Yes. So this is called lamellae. Okay. What are they called? Oh, why is this pen not working? Lamellae. And because they are present inside the stroma, in the space, they are called stroma lamellae. So these three terms, stroma lamellae, thylakoid membrane, and grana, are very, very. Let me write it as grana and draw two of them like this. Do you understand now? Grana, thylakoid membrane, and stroma lamellae. All are clear?
Yes.
Yes or no? Yeah. And they synthesize proteins and their own carbohydrates inside. So they can make sure, unlike mitochondria cannot produce its own nutrition inside. What it can produce is ATP. But for that, the raw material has to come from the cell. That was the deal between mitochondria and the eukaryotic cell, and they together became the animal cell. Plants evolved separately to animals. Okay. And plants has this additional benefit that in their cell, there is not only one alien, but two aliens surviving and living as the part of their cell, mitochondria and plastids. So they have a double benefit. That's why they ensured everything. They are the most dominant species on the planet without even moving a muscle. They don't even have a muscle. They don't move, they don't conquer, they don't make territories, they do not do anything. And they are the most flourishing and what do you say, successful living systems on the planet, plants, because they ensured their energy through mitochondria, they ensured their food production through plastids, they ensured their safety through cell wall, and they ensured their excretion and everything through a very big vacuole in the center, and they are living peacefully like saints. They just are at peace. No need to move, no need to work from home, no need to go office, nothing. Just put their roots in the ground, get raw materials, provide food for themselves, and also for people like also for species like us who are animals who cannot produce their own food. So we rely on plants. Like it? Yes, Iram. This is for plants, right? This emoji?
Yes.
Yes. So this is plant, exactly. Plants are in this mode. Ki you cannot compete with us. We are feeding you. We are feeding ourselves. We are feeding you. We are feeding the ecosystem. We are the boss here around. And then human goes and start cutting these plants for timber and making home, and starts doing deforestation and thinks that we can still save the planet. That is how fool we are, because we do not study biology to a detail where we realize that they are more advanced than us already. We are not, we may be the most intelligent species on the planet and the most versatile, but they are the most successful. They do not need anything. If all the plants disappeared from the planet, all animals will vanish. But if humans disappeared from planet, plants will not have any effect. They will be even happier. They're like, okay, gone. Good for them. Okay. So that's the strength and the beauty of a plant system. Okay. And apart from that, write down. It also, so yes, Aisha.
Then how would they get
Yes.
carbon dioxide?
There are other ways to produce carbon dioxide. When humans were not there, there was still carbon dioxide produced. Carbon dioxide is produced from other sources apart from humans. Most of the carbon dioxide is produced by zooplanktons. These are very tiny eukaryotes present in the ocean. There is a very good dynamic in the ocean. The biggest organism on the planet right now?
Blue whale.
Yes, blue whale can be up to 30 meters big, like a very big organism. So big that if a blue whale opens the mouth, you can just walk into the mouth, go and see all the stomach areas, move through the intestine like you are moving through a tunnel. You can even carry a small bicycle with you and then can come out very easily. But yes, this one. And what do blue whales eat? The biggest organism on the planet, what does it feed on?
Fishes in the ocean.
No.
Humans?
Blue whales. What? Humans? Then how are they alive? You're like, there is an association working for whales, sending humans, selecting humans and sending them to eat for the whales. No. Let me tell you a very interesting fact. The biggest organism on the planet cannot swallow anything bigger than this size, bigger than a berry or a marble. You can technically walk into the mouth of blue whale, but the blue whale will not eat you. It cannot swallow you. Blue whale can only feed on tiny microorganisms and crustaceans called zooplanktons and phytoplanktons. Yes, Aisha. There are many things in biology that we are not aware of, many wonders that no one is aware of if they do not understand biology. But this is the delicate balance. If whale started eating organisms, it can eat everything. And then the balance of the nature will not be there in the ocean. So that's why the biggest organisms feed on the smallest organisms in the ocean. And lot of CO2 comes from zooplanktons only. So even if humans are not there, do not worry about the CO2. Actually, we are producing more than what is required in the nature. That's why global warming is also ramping up. So plants are very happy without us. Don't worry about that. Okay, cool.
Sir,
Yes.
if humans go out of the earth, then who will give water to these plants?
Rains.
No.
Do you go and water all the forest plants?
No.
No one goes and waters all the forest. We just water the plants that we have in our terrace garden or around. Okay. There is a mechanism called rain. Rain will give water to all these plants. Okay. Very good. Good question though. Yes. No question is absurd in science. It's just every question helps you understand science better and better. Okay. Apart from this whole setup, this magnificent setup of producing their own food, they also have a small double-stranded circular DNA. Write down. So they also have ribosomes. Okay. Their own ribosomes. Again, these ribosomes. And one more thing. If the ribosomes are present in the plastid, so this is the structure of a plastid. It is not of a cell. Okay. this plastid is present in the cell, in the plant cell. You understand that, right? You're understanding that I'm making a plastid only.
Yes.
This is an organelle. Yeah, okay, very good. So they have their own ribosomes, which are 70S. Oh. Their own ribosomes are 70S, and the ribosome in the cytoplasm of the plant is 80S, because plant is a eukaryotic. They also have their own double-stranded circular DNA. Okay. Small circular DNA. So they are just like chloroplast, right? With an upgrade to be able to produce their own food. Yes or no?
Yes.
I sometimes wonder if instead of or along with mitochondria, animals also could have gotten plastids, at least the chloroplast. Things would have been sorted, no? Nothing. We would have done nothing. But again, we would have not done anything. We would just be lying in the sun and producing food, and we will not be moving a muscle to do all that thing that we are doing. You know, conquest, wars, jobs, scientific discoveries, you taking a biology class and giving an exam, all of that will not be there. So there's a very good belief, and actually there was an article I was reading in biology. Two things has driven animal evolution and has brought humans to the point where we are today. First is hunger, search for food, constant, because we cannot produce our own food in the body. We can cook our food. Once in a class, I said that humans cannot produce their own food, and one of the students said, but you know, my mother can. And I'm like, yes, you know, she can cook food. She cannot produce food. So that's the difference. Humans cannot produce their own food. We can cook our own food. That's a different thing. But he got offended actually. So that's why I'm just clarifying. So hunger, the constant conquest for food, has allowed us to be hunters, gatherers, and then later on we settled as agriculturers and we started agriculturing plants and animal livestock. So this is how we evolved. Second thing was the sexual drive. The sexual drive is the major drive that has generated variation. So we can do sexual reproduction, which can give rise to variations, different genes, which bring in different capabilities, enhanced capabilities, mutations, etc. So hunger is one of the very major reasons that we are so evolved. And plants are so settled, tension-free, but not evolved. You know, they do not have any second world, third world, first world problems. They just are rooted, they grow, they need sunlight, water, they are set. Okay, now draw this thing, please. Draw all of this, and please be clear about especially three terms: grana, thylakoid membrane, and stroma lamellae. Tell me when you are done drawing, both of you. And if you see, you know, still, whatever we all are doing, the conquest for food, you know, that is the baseline thing we all want to meet, you know, food. Whatever anyone do, if you are a scientist, a doctor, any profession you do, you're a bank manager, you're working for something, you're working to get food, right? Yes or no? It's all about hunger.
So,
Yes,
stroma
yes.
is Also a site for dark reaction.
Stroma is also a site for dark.
Dark,
Yes,
yeah.
the light and dark reactions I will take in photosynthesis when I will teach photosynthesis. I'm not taking it here. I'm just telling you about, yes, stroma is the site for dark reactions and thylakoid membrane is the site for light reactions. Correct. Okay.
What
So
is
just
dark?
okay. So you draw this first, then let me tell you photosynthesis here only. Okay. Just draw this, then I'll come to photosynthesis. I was thinking of taking photosynthesis. Do you have a plant chapter separately somewhere? Nutrition in plants or something like that? There should be, right? Because plant physiology is there in 11th, I don't know. Okay, let me tell you. It's a part of the connected knowledge, so no problem. Finish this fast, people.
There is photosynthesis in higher plants. That one?
Oh, there is a chapter, right? Photosynthesis in higher plants.
Yes.
Great. So there I will be telling you about. So let me just, for your curiosity, let me just tell you verbally, and I'll take this part in detail there. Okay, in photosynthesis chapter. So what happens in photosynthesis is, we know that in photosynthesis, the first step is to harness the light energy, right? That light energy is converted into chemical energy and then used for chemical reactions in the cell. You understand that, Iram? That part is clear?
Yes.
Yes. But what during the night? Because sun and the light is only available during the day, right? That is, so photosynthesis is divided into two parts. It has light reaction and it has dark reaction. Now the word dark reaction is a misnomer because scientists first discovered the light reactions, the part that only happens in the presence of light, which is harnessing the light energy, converting it into the chemical energy. But once it is converted in the chemical energy, you no longer need light, right? So processes from there can happen independent of light. So scientists name those processes as dark reactions, but it's not that it only happens in the dark during the night. It happens both in the day and in the night, but it is free from light. It is independent of light energy. So it should be light energy, or a better name according to me should have been light dependent reactions and light independent reactions. But scientists, when they named it light reactions and dark reactions. So dark reaction is where the food is produced, actually. The starch or the carbohydrate is synthesized. But that happens during the day also. Okay?
Okay.
So the light reaction, which is dependent on light, happens in thylakoid membrane because they contains chlorophyll. That's why I have made them green. But the dark reaction, which is once you have energy in the cell in the form of chemical, you no longer need to just stick to thylakoid membrane. The other reactions can happen anywhere in the stroma. So stroma is just like the cell equivalent to a cell cytoplasm. So this is the fluid of the plastid only. So the space covered by the inner membrane, whatever is inside, is called stroma. Is it clear, Iram? So
Yes,
the light independent reaction can happen anywhere, right? It can happen anywhere in the stroma. So stroma is the site for dark reactions, and thylakoid membrane is the site for light reactions. But we'll do it in detail, and it's very interesting in photosynthesis chapter. Okay. Cool. Very good. Is this done? Drawing is done. Shall I go forward?
Yes, done.
Okay. So write down. Should I dictate everything that I just told and draw, or can I rely that you can make notes on the basis of this, written notes on yourself? I want verbal confirmation. Can you do it, Aisha?
yeah, we can do it.
Yeah. So go through the NCERT. I think the NCERT is very thorough with this plastid thing. and make notes, written notes. So I told you about types, and I also told you about the structure. Okay, some important things that you should know. Yeah, I told you it has smaller circular DNA, ribosomes, stroma, lamellae, inner membrane, outer membrane, everything. Okay, cool. So this was plastid. Okay, this is another. Plastids were also, they also came into a plant system through endosymbiosis, because they also are like a photosynthesizing bacteria. You know, there are many bacteria that can photosynthesize.
Yes.
Have you heard of a term called blue-green algae, BGA?
Yes.
You know, it is not an algae. The name is again a misnomer. Blue-green algae is a bacteria. It's a cyanobacteria. But when it was first discovered, that they found out that it can photosynthesize. Oh, then it must be an algae, and it looks bluish green. So name it blue-green algae. But it actually is a cyanobacteria. Okay, it can do photosynthesis. So similarly, this plastid resembles a prokaryotic organism which can do photosynthesis. Yes. Yeah. And mitochondria resembles like a prokaryote which can do aerobic respiration. So inclusion of both in a eukaryotic cell is an advantage to the eukaryotic cell. Plants have both, so they are at a higher advantage from survival point of view and from success point of view. Animals just have mitochondria and not plastids. Very well. Now, apart from these, the next thing that we will come to, so we have finished mitochondria, plastids, both. Okay, ribosomes we have already done, right? We have done ribosomes, you know.
Yes, we have done it.
Yeah, we have done. Yeah, you only told about it. So now from the endomembrane system and the non-endomembrane system, let's come to something which is called cytoskeletal system or cytoskeleton. Now, what does the word cyto means? Cyto, cell. And what does the word skeleton means? Skeleton means the rigid support that gives shape to any system, right? That's skeleton. For us, our skeleton is made up of bones, which is basically calcium carbonate based, and it is inside the body. So that's why it's called endoskeleton. It's inside. We cannot see it from the outside. For an insect, I told you they have an exoskeleton, right? The thing that gives shape to their body is present on the outside, made up of chitin. Yes or no? Just some brush ups.
Yes.
Yes. And if you crush that exoskeleton, the organism will die. Similarly, a cell also has a skeleton. Something gives shape and size to a cell. A cell is not like a jelly all the time. All cells are not jelly or squishy. You know, some cells maintain their shape, and that is because of the now when I talk about cytoskeleton, I'm not talking about the cell wall. Cell wall is for protection. Okay, outermost. It limits the growth of the cell. So whatever will be the size and the shape of the cell wall, the cell will have the same shape and size. But inside the cell, there are certain elements that make a network and give the cell a rigid size and shape. Okay. So write down. Cytoskeleton is an elaborate network. Cytoskeleton is an elaborate network. Now, though we have not finished this chapter, but let me ask you this question. We are almost like 70%, 60% in the chapter. Do you realize that we are just studying about a single cell from last 3 classes? And do you realize that a single cell is not as simple as we think it is? It is a world in itself. Yes or no? It has its own endomembranous system. It has its own powerhouse. It has its own setup to produce food from light. You know, all these things it can do. Then we are now going to talk about the cytoskeleton. Yeah. Then we'll be talking about the nucleus, which is there, which gives rise to all the genes and all the proteins. So do you see, it's a micro environment. It's like a country being run, and the king is sitting in the nucleus, the prime minister is sitting in the nucleus as the form of the DNA. And we have other associates which entered the cell and started living as an organelle, and brought some additional. They're like our, what do we call it? Then you conquer some nearby country and brought this country to and your country expanded and you now have more resources. So those are mitochondria and plastids. But do you understand how complex even a cell is in biology? And we have not yet gone to the tissue level or organ level or organ system level or organism level. So as a doctor, when you will become a doctor or anything related to biologists, Aisha, no, Aisha, but if you understand the fundamental, you are going to know and help a lot, right? Yes, no pressures. I'm just telling you to appreciate the magnificence of the system so that you can proudly say that, I study biology. It's not an easy peasy thing to do. If it was easier, everyone would have done it. It's something that I am doing. So just have that feeling when you're studying biology, science in general. Okay. Now write down. I think Iram is saying enough of pep talk. Complete the definition, please. Right, Iram? Okay, write down. Cytoskeleton is an elaborate network of proteinaceous structures. Which means it is made up of protein. That's why we call protein as the fundamental structural unit of cell. So elaborate network of proteinaceous filamentous. They are like filament. It's exactly the same like when we make a house or a building, we first put the foundation. For that we use the bars, the iron bars, right? What do you call it? The iron bar. You understand what I'm talking about? Around which we put the cement and then we fill it with the bricks. I hope you got it, right? Iram, Aisha?
No, sir. Could you tell again?
Yeah, when we make a building, we put some filamentous iron bars first, like this. And then around it, we make a pillar, right? Then there are multiple such pillars, and we fill the gap with bricks to make a wall. But there has to be multiple pillars in a building, load-bearing pillars, right? And even for the roof, we make a mesh network of these iron bars. You understand what I'm talking about now? The roof and the floor and the walls, how they are made,
Yes.
There are strong iron bars going through and through inside, around which there is concrete and cement and everything put, right? So the filamentous structure of these cytoskeletal networks is also like this iron bars. So that, it is an elaborate network of proteinaceous filamentous structures which includes, so there are 3 types of cytoskeletal structures, which includes, the first one is called microtubules, which includes microtubules, then microfilaments. It's also known as actin filaments. Okay. And the third thing is intermediate filaments. Intermediate filaments. So these are thin bars or structures of protein which gives shape, size, and support to the cell from inside. Okay. So collectively, all these three, microtubules, microfilaments, and intermediate filaments, collectively are called cytoskeletal elements. Cytoskeletal elements. Now write down what is the function, what is the need of this. So the first is to provide mechanical support from within the cell. From outside, it is cell wall that provides mechanical support, right? If the pressure comes from outside, the first thing is the cell wall. But in a human cell also, in an animal cell also, there is some sort of shape. In the very beginning of the chapter, we talked about different types of cells in the human body. Remember, Iram? I think Aisha was not there in that class, but we discussed about neuron cells, muscle cells, fat cells, RBCs, right? RBC has a particular shape. WBCs which do not have a shape. Remember all these things? So that shape is maintained through these cytoskeletal elements. So first is mechanical support from within. Yes. Yes,
These
Aisha.
cytoskeletons are present in every cell.
In every cell, yes, they are present in every cell. There cannot be a cell without cytoskeletal elements. Okay.
Yes.
Provide mechanical support.
So they are also in prokaryotic cell?
Yes, they are also in prokaryotic cells. Okay. A cell can be without a nucleus but not without a cytoskeletal elements. Because this is what forms the cell. This is what makes the cell. Then you can have certain organelles or you cannot have certain organelles because the organelles bring functionality. You understand? For example, you need to make a room, a house. Inside the house, if you do not have a refrigerator, it's not like you will not have a house. It's just like you will not have access to some things. If you do not have a television or you do not have something which helps you in different functions. You understand? So organelles are like which makes the life of the cell or functionality of the cell easier and diverse. If you have mitochondria, don't worry about energy. But if you do not have mitochondria, if you are a bacteria, then you have to worry about energy. Then you have to constantly find food sources or find other sources from where you can get energy. Similarly, but whether it is a prokaryotic cell or a eukaryotic cell, there has to be a structure, right? And that structure will come from cytoskeleton. Do you understand, Iram, Aisha?
Yes. Sir, where
Yeah.
is it present? It's like it's present in cytoplasm, plasma.
Yeah, it's present, yes. So the way we imagine, I will try to show you a video if that video does not have a copyright issue. There are many things I can show you in class through videos, but many things have copyright issues if I just run in the class because it's also being recorded. So the point is you can go to YouTube and look for cytoskeletal elements or internal world, inner world of cells, things like that. And you will see the way we imagine a cell, that there will be a membrane like a balloon and inside there will be an empty space. It's not like that. A cell does not have like empty space. It has something running from here to there, some intermediate filaments connecting the nucleus to the membrane to the Golgi to the ER, things like that. All of the cell is jam packed like a very, very busy city, of Shanghai or New Delhi or Dubai. It's all crowded. You understood? So it's present everywhere. Yeah. Second, write down, it helps in motility of the cell because cytoskeletal elements form the flagella. Flagella are formed from these microfilaments and intermediate filaments. Okay. And third, maintain the shape of the cell. Maintain the shape of the cell. Now we will go into the detail of it through cilia and flagella. We will be understanding about the nature of intermediate filaments and microfilaments through cilia and flagella. And in the next class, we will do centrosome, centrioles, cilia, flagella, nucleus. I think these are the only things left. And in the nucleus, I will spend some more time in explaining DNA, chromosome, because that is very, very important concept. And then we'll end this chapter. And once we understood cell, then we will go to the new chapter where we will start dividing it, how it divides. So that is a whole new world where the cell divides. We'll start the action there. So I think so let me stop here for today at the introduction of cytoskeleton. If you have any questions, please ask. Anything? Any doubts?
No, sir.
No?
Sir, I
Okay,
have a
so
question.
yes, please.
Sir, I have a question regarding the cell wall.
Mhm.
So, above the cell wall, there is also a layer called Glycocalyx. Is it the same as lipopolysaccharide?
No, first of all, Glycocalyx is limited to certain organisms, and Glycocalyx is basically it's made up of the word glyco comes for sugar, so it is made up of proteoglycans or glycolipids. It is a network basically. More than you will say it's a supportive layer above the cell wall. It is in connection with the cell wall only, but you can say it is a cell coat. There is another word we use cell membrane. Then above cell membrane we say it is cell wall, and in some organisms we say that there is a cell coat. So it is just an extension of the cell wall only, but because it is not present in all cell wall containing organisms, so we use another term called cell coat. You understand? Is it clear?
Yes.
Yes. So it is a network of polysaccharide instead of lipopolysaccharide. Lipopolysaccharide is more smooth. Polysaccharide is carbohydrate based. So it does not have a lipid component. But yes, if it is present, it will provide, of course, it will provide some more protective advantage to the cell.
Okay.
Okay. Cool. Anything else?